As you know, the PDBP biomarker program for Parkinson's began in 2012. And the idea was to fund multiple different investigators with their best ideas to develop discovery projects around really promising biomarkers. But in addition, the group came together to standardize how they collect their data and to put their data, their clinical data, as well as biosample data in a repository. Now, after years of collection and the cooperation of patients and investigators, the NINDS and the Michael J. Fox Foundation have put together a really very interesting, very rich data set. And this brought in the interest of industry partners who are also focused on developing therapies for Parkinson's disease. And so they come together to make the most of these rich biosample and clinical data and analyze them to try and uncover what secrets there are in this data that would help the common goal of both the industry and the academics to develop better treatments for Parkinson's disease. So without the Parkinson's Disease Biomarker Project, I don't think we'd be in this really exciting spot where we are now with industry, academia, and the government all working together with this tremendously rich data set and biosample repository to uncover these secret biomarkers that are so essential for developing treatments for Parkinson's. So at NINDS, we are the institute that's the major funder of research in Parkinson's disease. We have learned a tremendous amount about Parkinson's disease in the last 20 to 30 years. And much of this has come from genetic studies that have identified, say, synuclein as the protein that aggregates in cells that then die in patients with Parkinson's disease. And we think that this is a major cause of their symptoms. We are learning now that synuclein likely spreads through the nervous system from one neuron to another. And these discoveries certainly make people think, well, how could we slow this disease down? How can we stop the synuclein from aggregating? How can we stop the synuclein from traveling in the nervous system? How do we stop the synuclein aggregation from killing the cells? But in a patient with Parkinson's disease, you can't answer those questions now because we can't see the synuclein. We can't see its effects either as it moves through the nervous system or as it affects the cells until, as we think in Parkinson's disease, 80% of the neurons are dead already in the nigra before the patient has symptoms. So we desperately need these biomarkers to be able to track these different stages of the disease. The real holy grail for Parkinson's disease is to have a biomarker that can detect these changes and we can intervene before the disease even causes symptoms. Because the other area which is really exciting in Parkinson's disease is the data that the process is going on at least 10 and maybe longer than 10 years prior to the development of symptoms. So if we have a good marker, we think we can intervene early. We could potentially cure the disease by stopping its progression before it causes any symptoms. So that's the ultimate goal, I think, of the biomarker program. So as you know, the Parkinson's Disease Biomarker Program began in 2012, so that was 6 years ago. And people have been working very hard in trying to discover biomarkers that would be helpful in developing new treatments. But they have also been contributing to a massive effort to bring together samples and clinical data from large numbers of patients. And this we've done in collaboration, particularly with the Michael J. Fox Foundation. But this has led to a repository of clinical biofluids such as blood, serum, plasma, spinal fluid on thousands of patients. And the efforts in the PDBP attracted the interest of many private partners, including industry partners, and we formed what's called an Accelerated Medicine Partnership, which is a standard NIH public-private partnership method to utilize these samples and to analyze these samples in depth to see what kind of information is there that can inform all our common goal, which is to find a better treatment for Parkinson's disease. So without the 6 years of efforts on the part of the investigators and the staff in the Parkinson's Disease Biomarker Program, I don't think we would be where we are now with this really exciting public-private partnership which has a singular focus to develop biomarkers that will inform how to better develop treatments for Parkinson's disease. So the National Action Plan for Alzheimer's Disease has a roadmap of how we in the US, and particularly through the research at NIH, can try and make a dent in the growing public health problem of dementia in this country as our population ages. Interestingly, and very important for us, the NAPA plan includes Lewy body dementia as one of the dementias that are receiving this additional funding on behalf of from the Congress and the taxpayers. And we believe that we, by incorporating the work of investigators understanding how synuclein spreads through the cortex and causes dementia in patients with Parkinson's or with Lewy body disease, where the dementia comes first, Parkinson's comes later, that these two efforts of the PDBP and the Lewy body disease work can really come together in a way in which it could not happen had it not been for the Parkinson's disease biomarker program. So much of what we're doing now with this additional funding is kind of bringing the Parkinson's disease biomarker program to the Lewy body dementia field. And really, really important. But it is a very difficult situation. As you know, in Parkinson's disease, the patients have very good cognitive function, and they're so interested in volunteering to make a difference in the disease. Much harder as people develop cognitive changes, as occurs in Lewy body dementia, Lewy body disease. So we need really the help of the caregivers, patients with Parkinson's, and the general scientific community to help us recruit patients with Lewy body dementia to serve as a complement to what we've done in Parkinson's disease.